Hello, and welcome to this lecture on sepsis and septic shock pathophysiology. My name is Andrew Conway Morris. I'm a clinical academic in intensive care at the University of Cambridge and at Addenbrooke's Hospital in Cambridge. And my research and scientific interests are in the immunology of critical illness, including sepsis. These are my conflicts of interest. So, the first thing to say is that to understand this lecture requires just a undergraduate understanding of immunology. So it's not going to go into too great detail, so don't panic. The first thing we'll talk about is the importance of the pathogen and the site of infection and how this can lead to an inflammatory response. We'll then talk about the early tissue immune responses to pathogens and how some of those signaling events can become systemic, which leads on to how that inflammation spills over, and so how we get from systemic signaling to systemic inflammation, which is of course one of the hallmarks of sepsis and septic shock. We'll talk about the mechanisms by which organ failure occurs, and then we'll talk a bit about a specific organ failure, which is a failure of the immune system, which is often found in patients with severe infections, and the implications of that failure. And finally, we'll talk about the self-reinforcing cycles that can occur that lead to multi-organ failure and ultimately to the patient either needing to be admitted to ICU or, unfortunately, to their demise. Now, before we get started, it's important to remember the definitions of sepsis. So, as you, I'm sure, are all aware, sepsis is organ failure resulting from a disordered immune response to infection. But of course, the order that this thing happens in is the reverse. We start with an infection, which produces a disordered immune response, which results in organ failure. So let's talk about the organism, the infecting organism. So there are some microbes which are simply not pathogenic. They do not cause disease in certainly immunocompetent adults. However, there are other organisms which can be pathogenic depending on where you find them. So if we take, for example, Staphylococcus aureus, that can live quite happily in your nose without causing any problems. And indeed, about a third of us will be permanently colonized with this organism with no apparent ill effects. However, if that same organism gets into another space, in this case the lungs, it can and does cause a severe infection, in this case staphylococcal pneumonia. To give another example of yeasts, if we take Candida, they live quite happily in your gut and don't cause any problems. If you get candidal overgrowth in the lungs, as may happen, patients who are on broad-spectrum antibiotics, again, the candida can live there quite happily without causing a pneumonia. However, if that candida gets into the bloodstream or into the peritoneal cavity, it can cause serious illness. It should also be noted that some bacteria produce specific toxins. An example would be pantine valone leukocidin, or PVL toxin, which is carried by some Staphylococcus, and staphylococcal and streptococcal toxic shock toxins. However, these are the minority of cases. So most cases of sepsis arise from organisms that don't produce specific secreted toxins. It should also be noted that although bacteremia is associated with worse outcomes in sepsis, it is not necessary to have bacteremia in order to develop the organ failure, which we'd call sepsis. So the first place that the organism, the host, tends to encounter pathogens is in the context of an epithelial surface, often in the luminal organs, so those mucosally lined organs such as the lungs or urinary tract. And at the mucosal surface, there are a number of defenses against pathogens. These include luminal immune cells, particularly macrophages, mucosal immunoglobulins, immunoglobulin A, if the patient has already encountered that pathogen's patterns before. And you may also see tissue immune cells starting to sample the luminal surface. So this is a dendritic cell, which has poked out one of its dendrites in a process called snorkeling, where it samples the luminal aspect from within the tissues. And of course, there are other defenses as well. We have the epithelium itself and often a lining of mucus. If, however, the pathogens are able to penetrate these defenses or even overgrow within the luminal space itself, we end up with infection, which is a host response to an invading or overgrowing pathogen. And here again, we have similar tissue immune cells to those found in the lumen, so macrophage here and dendritic cells, which encounter the pathogens. And through pattern recognition receptors, so these are receptors which are designed to recognize broad microbial patterns rather than being specific to an individual organism. And they recognize these patterns. So for example, here we have lipopolysaccharide being recognized by toll-like receptor 4, or flagellin, a component of the flagella of flagellated bacteria, ligating toll-like receptor 5. And when these receptors are engaged by their target, this triggers a series of intracellular signaling pathways. This includes the formation of the inflammasomes, activation of caspase 1, and it exists situation here, cleavage of interleukin 1 beta from its proform into its active form, which is then secreted. As well as working with pre-existing or pre-stored proteins, these signaling pathways also trigger de novo protein synthesis. So here we can see pro-inflammatory cytokines being released, and depending on which pathways are activated and which receptors are ligated, we also get release of interferons, for instance, type 1 interferons. And these new products, as well as these stored products, are released into the extracellular environment, and they essentially sound the alarm signal. They say to the other immune cells in the local tissue, but also more widely into the systemic circulation, there's a problem here, and we need some help combating this infection. So what we see is activation of local immune cells. We then start to see this self-reinforcing cycle of inflammation, as well as recruitment of circulating immune cells. We also see the release of vasoactive compounds, which lead to vasodilatation, increasing local blood flow to the area. And we see the loosening of the tight junctions between endothelial cells, which facilitates the transport of, or movement, migration of immune cells, particularly neutrophils, from the circulation into the tissue spaces, where they can start effecting antibacterial defenses. Several mechanisms of antibacterial defense exist, or antimicrobial defense exist, including phagocytosis, the ingestion of particles, release of granule contents with lytic enzymes, reactive oxygen species generation, and in some cases the release of so-called neutrophil extracellular traps, which are DNA-containing traps which can trap and kill bacteria. So these responses all produce the characteristic heat, swelling, and pain that is associated with inflammation. Now, just to touch a little bit on phagocytosis, which is a major way by which neutrophils, certainly, and macrophages kill bacteria. So phagocytosis is the process of ingestion and killing of bacteria. So here we can see a neutrophil, which is encountering a target, in this case a yeast target, and you can see that it has ingested it. Here we can see a neutrophil, which is when exposed to Staphylococcus aureus, here in fluorescent green. These Staph aureus are ingested. And then if we add a pH-sensitive dye, you can see that the phagosomes become acidified over time. So the longer the neutrophil has those particles in it, the more it acidifies its phagosome, which is part of the killing mechanisms. And here we can see the outside of the phagosome outlined by a phagosome surface marker. So this is an important mechanism by which neutrophils and other cells, other immune cells, can kill bacteria. So that's the sort of local tissue response to infection. But what happens to make this systemic? Because, of course, that's important. So we've already discussed these processes here, whereby tissue immune cells and recruited immune cells start to kill bacteria, releasing inflammatory cytokines and chemokines, some of which get out into the systemic circulation or into the interstitial spaces where they're then drained by the lymphatics. And these pro-inflammatory mediators will activate the immune system at distal sites. We also see damage to the tissues at the sites of infection, which releases so-called damage-associated molecular patterns. And these are rather similar to the pathogen-associated molecular patterns that ligate the pattern recognition receptors that we mentioned earlier. So we get damage at a distal site, activation of the immune system, and certainly in the case of the lungs, these activated neutrophils will get stuck in the lungs and then migrate in and produce an inflammatory response at a distant site, so-called indirect acute respiratory distress syndrome, which is a common feature of sepsis. It also has to be said, however, that this immune cell infiltration is not the only mechanism by which we see organ damage in sepsis and septic shock. Now, the systemic response to a severe infection, so an infection that can't just be handled by the local tissue response, is, at least initially, an adaptive response, and the bone marrow plays a key role in that adaptive response. So what we see is an early release of stored neutrophils from the bone marrow, and then if the demand continues, we end up with a process known as emergency granulopoiesis. And this is where the bone marrow shifts its production from other white blood cells, such as lymphocytes or other granulocytes, into predominantly producing neutrophils, which are then moved out of the bone marrow quite fast. And if the stress is particularly marked, then you see immature or band-type neutrophils in the circulation, and sometimes even myelocytes and promyelocytes in the circulation, depending on the degree of stress. And this is, at least in its early forms, an adaptive response with the body trying to ramp up its ability to fight bacteria. However, as we well know, this can become maladaptive. And the question really is how that happens. Well, there's a whole range of mechanisms, but many of them involve alterations in tissue oxygen delivery and tissue oxygen handling. So one of the prominent features that we see in patients with sepsis is mitochondrial dysfunction. And this mitochondrial dysfunction is driven by a range of different mediators, including cytokines, catecholamines, both endogenous and those that we give patients, and also antibiotics. Mitochondria are originally derived from bacteria and are susceptible to toxicity from antibiotics. So we see mitochondrial dysfunction and an inability to handle the oxygen that the cells are having delivered to them in a wide range of cells, even if there isn't overt immune cell infiltration. We also see changes in the microvascular. So this is some pictures from Daniel DeBacca's study from 20 years ago now, showing marked heterogeneity in the microvascular blood flow. So you can see here some blood vessels have got almost no flow at all, while others have really quite brisk blood flow. And the reasons for this microvascular dysfunction are multiple. We see microthrombi forming from immunothrombosis. We see piling up of white blood cells blocking small blood vessels. And obviously we've got diversion of blood away from core organs due to vasodilatation. So all of this can impair tissue oxygen delivery to cells which already can't handle it due to the mitochondrial dysfunction that we've mentioned. And of course, especially in the lungs, the inflammatory infiltrate itself, both with the fluid and the cells, can impair organ function. So in the case of the lungs, impairing gas exchange, which of course is one of the lung's core functions. So all of these processes can lead to organ failure, which is of course the hallmark of sepsis. And one of the most important organs that we have is one that is perhaps not immediately thought of as an organ, but is the immune system. And in common with other organ dysfunction, we also see immune dysfunction. And what's interesting here is that some of this dysfunction is driven by pro-inflammatory mediators itself. So, for instance, C5a, which is a component of the complement cascade, which is released in large amounts in systemic inflammation, impairs phagocytosis, but also, in relatively recent work, has been shown to impair phagosomal maturation. So here's an example of a video. So here we can see neutrophils similar to the ones that I showed previously, but these have been exposed to high concentrations of this complement component C5a, and you can see here that they are able to move. They're not completely paralyzed, but are unable to ingest the particles that they encounter. And of course, this is associated with an increased risk of secondary infections, as well as failure to clear the primary infection. Now, immune dysfunction occurs across the range of immune cells. There's almost no immune cell that's been examined in the context of sepsis in which dysfunction hasn't been identified in one study or another. And this dysfunction occurs across both the innate immune system and the adaptive immune system. These dysfunctions don't all occur simultaneously. There are degrees of dysfunction, but the greater the number of cells that are affected, the worse the outcomes are for the patient. Now, the other thing is that, of course, the development of organ failure is not simply influenced by the organism and the site of infection. There are other factors that influence organ failure. So certainly time to effective therapy. If there's effective therapy given early, then sepsis and organ failure are less likely to develop. Of course, patients who are at the extremes of age, patients who are frail, are more likely to develop organ failure. And that is in large part because of the association between age and frailty and chronic organ failure and comorbid illness, such as immunosuppressing conditions. And of course, one of the most important factors that determines whether a patient develops a maladaptive response is their genetic predisposition, which is the single biggest predictor of adverse outcomes from infections. And of course, finally, we should remember that the organ support systems that we use ourselves can also damage the organs if used injudiciously. So, for instance, ventilator-associated lung injury, and of course, the breaching of mucosal defences that is necessitated by organ support, which can increase your risk of secondary infections. So, we've described the initial tissue response to infection, how that can become systemic, and some of the mechanisms by which organs fail. And really, I think what we need to do is think of this as maladaptive cycles. So, for instance, we have an initial bacterial response. We get release of pro-inflammatory mediators, complement activation, cytokines, nitric oxide, vasodilatation. This leads to cellular dysfunction, cellular damage, release of these damage-associated molecular patterns, which leads to further release of pro-inflammatory mediators, immune activation, which, as we've just mentioned, can drive immune failure, leading to persistence of existing bacteria or development of new bacterial infections. And so we get locked in this vicious cycle. Now, this is only one of the vicious cycles. There's another example here. We have cellular hypoxia for mechanisms we've already described, which can lead to dysfunction of endothelial cells and cardiac myocytes. This leads to shock, both from vasoplegia and cardiogenic shock. That contributes to heterogeneous microvascular flow, which leads to microthrombus formation, worsening cellular hypoxia. And again, we get locked into a vicious cycle. Now, of course, these cycles aren't separate. They interact with each other. So cellular hypoxia leads to cellular damage, release of further molecular patterns, further driving the release of pro-inflammatory mediators. Complement itself can directly activate coagulation, leading to further formations of microthrombi. Bacteria can also drive coagulation. And so we get these interacting, self-reinforcing cycles where even if you remove the ultimate sort of precipitating cause with effective antibiotic therapy, once this has been set up, the organ failure can persist after the infection has gone. And this is what we see often in critically ill patients who've been in intensive care for more than a few days. So, to summarize, Infection severity is determined by the pathogen, the site of infection, the site of where it's encountered, and of course the host response. If the local tissue response is insufficient, it will drive a systemic response which is aimed at clearing the pathogen, so initially an adaptive response. However, this can spill over into a dysfunctional hyperinflammatory situation where both local and systemic inflammatory activation drives organ failure. The reasons for this transition are not fully understood, but involve many of the factors that we've discussed today. There are multiple mechanisms that underpin this organ failure, but importantly, impaired antimicrobial functions of immune cells are also seen, and these are often driven by the same inflammatory mediators that drive organ failure. So putting us into a sort of worst of all possible worlds scenario with both systemic inflammation, immune-mediated organ damage, and an inability to clear primary or even secondary bacterial insults. And these vicious cycles set up driving further organ failure, further immune impairment, which, if left unchecked, will lead ultimately to the patient's demise. So thank you very much for listening. If you have any questions, please do feel free to reach out via any of these channels here, and I will do my best to answer any questions that you have. Thank you very much.